All right, we're going to go ahead and get started. So first of all, my name is Eric Morgan, and I'm a software engineer at Mindware Technologies, and I'm going to be your host today for this webinar on the heart rate variability analysis application. And if you've been a part of one of our webinars before, you'll know that it's typically segmented into main portions. There's the walkthrough, where we're going to talk about the application, followed by a question and answer session at the end. For today's walkthrough, we're going to start by giving you an introduction to heart rate variability, what it is, what it can do for you, and the signals that are needed to investigate it. We're going to talk about the processing techniques by which we obtain meaningful results from heart rate variability. And then we're going to jump into the application, and I'm going to show you how to actually get those results within the application. And we're going to talk about the ECG editing techniques for heart rate variability, how we're going to edit our data to make it acceptable for heart rate variability, which is a little different than if you're editing ECG data for, say, impedance cardiography. And finally, I'm going to show you how to output all of those results to an Excel spreadsheet for import into SPSS or other statistical processing packages. And so following that webinar, or following that walkthrough, we're going to be doing a question and answer portion where I'll be answering any and all questions that you bring up throughout the webinar. And you can do that by using the GoToWebinar interface and just entering a question here. So we are going to hold off on answering the questions until the very end of the webinar during that Q&A portion, especially if those questions are going to be valuable for the group to hear the answer to. So if you ask questions, and I encourage you to ask questions throughout the webinar, but don't be discouraged if you don't get an answer right away, because we will see it and we will eventually get to it. So we're going to start by talking about the signals that you need to acquire in the first place to study heart rate variability. And the first of those is the ECG, the electrocardiogram, or sometimes referred to as EKG. And this is a critical signal for doing heart rate variability analysis. And the reason this is so critical is because we can obtain precise beat-to-beat timing by detecting the R peak of the ECG signal. And the R peak, if you're familiar with an EKG signal, is the sharp peak that's present when you collect the signal using a lead-to configuration, which is what we typically recommend and what we have shown on our electrode placement poster that we provide. And because this R peak is so pronounced, it's so easily identifiable, it's a great landmark to use when determining beat-to-beat timing, which is essentially what we're studying when we look at heart rate variability. And if you'd like to learn more about the ECG signal, the different ways that it's processed, the different landmarks on it, we have a blog series titled All About ECG on our support site, support.mindwaretech.com, that goes into depth about all things ECG, everything you'd want to know about the signal, its origins, and how it's used. So that is the first key signal that is needed for heart rate variability analysis. The second one is respiration. And contrary to popular belief, respiration is not used directly in the calculation of RSA, or respiratory sinus arrhythmia, which is an important statistic when we're looking at heart rate variability. And we're going to talk a little more about that in a few minutes as to what that is. So respiration is not used in any of the calculations that people report on for heart rate variability. But what it is used for is validation. So respiratory sinus arrhythmia obviously involves respiration in some way. And without recording respiration to see that the respiration falls within an expected frequency, we can't say for sure that your RSA measure is valid. So if you're planning on reporting RSA, it's very important to collect respiration as well. And respiration can be obtained from a respiration belt, or it can be derived from the cardiac impedance signals, such as Z0 or DZDT. And this is particularly powerful when you do it in this way, because you can start to look at both branches of the autonomic nervous system, both sympathetic and parasympathetic, with just collecting ECG and cardiac impedance and then deriving respiration from that cardiac impedance. So definitely something to think about when you're getting set up to collect data, what you can do ultimately with the signals that you're collecting. And if you're not familiar, respiration is this sort of sinusoidal waveform of inspiration followed by expiration, or breathing in followed by breathing out. And typically, when the signal is increasing in amplitude, that correlates to inspiration, and when it falls, it correlates to expiration. But again, this is a little bit equipment dependent, so it may not be exactly that, but the cycles are going to appear like this. Some other signals to consider when getting ready to study heart rate variability are the plethysmograph, sometimes referred to as PLE or PPG. And this would not be used alongside ECG, but rather as a replacement to determine beat-to-beat timing. The problem with this signal is it's not ideal for obtaining precise beat-to-beat timing. And that's because the peak of the PLE is more rounded. So it's not always obvious precisely where the peak is. And when we're studying heart rate variability, we're looking at things in terms of milliseconds. And the rounded peaks on a PLE signal, there could be millisecond differences in where the peak is actually placed. So that's significant when we're looking at heart rate variability. And the other problems are sort of related to noise. So if you are collecting PLE using a finger cuff or an ear sensor, there's a lot of movement, especially when collecting it on the hand, that could be coupled into your data, resulting in a lot more time spent doing editing. So something to keep in mind if you're planning on studying HRV with a PLE signal. Another way that we can study HRV in the HRV analysis application with Mindware is through direct IBI series import. So if your device that you're collecting it with, maybe a watch or something like that, that's not able to record ECG, but is only able to record beat-to-beat timing in a spreadsheet or something, we can import that still and work directly from that series to obtain all of the meaningful statistics. The problem with doing that is you lose your ability to edit the series. So whatever came out of your instrument is what ultimately is going to be used to determine the statistics, regardless of whether or not there's artifact or error in it. And if you're interested in doing this sort of thing, if you're working with a device that only outputs an IBI series, you can check out this knowledge base article number 23 on our support site, and that'll tell you how to import that data into the HRV application. For this particular webinar, we're going to assume that the ECG signal was collected, and respiration as well will mostly be derived from the cardiac impedance signals. So those are the different signals that can be used to look at heart rate variability. And now we'll talk a little bit about what heart rate variability is and why it matters. So obviously HRV stands for heart rate variability. And what we're doing is we're measuring the variability or the change in timing between successive heartbeats. And we refer to this time between heartbeats as the interbeat interval, or IBI. And you'll hear that acronym mentioned many times throughout this presentation. And these changes in IBI can occur due to autonomic nervous system regulation of the sinoatrial node of the heart, which is where the beat originates. And we'll take a step back real quick and just talk about the autonomic nervous system, which is comprised of two branches, the parasympathetic and the sympathetic branches. The sympathetic branch, abbreviated as SNS, is considered the fight or flight branch. So it's these quick reactive types of regulations. And then we have the parasympathetic branch, which is the rest and digest. It's sort of the slower moving regulatory mechanisms of the autonomic nervous system. And it's been shown that heart rate and even HRV are influenced in different ways by both branches of the autonomic nervous system. But something that's really powerful when studying HRV is the ability to isolate one of the branches of the autonomic nervous system so you can really understand how it's influencing regulation in the body. And that is done by isolating a specific frequency of variability that has been proven to be indicative of parasympathetic nervous system influence. And that's the high frequency heart rate variability. This brings us to our next topic, and a word or a phenomena that I already alluded to earlier, which is respiratory sinus arrhythmia, or RSA. And what this is it's the physiological phenomena by which your heart rhythm or beat-to-beat variability is influenced by your respiration. And typically, this is the most reported on index for high-frequency HRV, and therefore it's indicative of parasympathetic nervous system influence. And in Mindware, we use the frequency domain analysis technique to obtain it. And essentially, what we're seeing here is heart rate increasing during inhalation and decreasing during exhalation. So what we see here is the ECG signal with a respiration waveform overlaid. And again, this is a bit of an artistic rendition. Typically, the transition from high heart rate to low heart rate would be more gradual. But suffice it to say that when you're breathing in, the heart increases, and when you're breathing out, the heart rate decreases. Some other measures that are interesting from heart rate variability perspective are some different frequencies. So we can look at not only the high frequency or RSA specific variability, but we can look at low frequency, which was once believed to be indicative of sympathetic nervous system influence, but more recent studies have shown that it's actually influenced by both branches, so sort of up in the air as to how that pertains to autonomic balance. And then we have the very low frequency, which is not as well defined and does really require further assessment, but it's most commonly used in a clinical domain. And beyond the frequency domain measures, there are a whole series of time domain statistics, which are entirely based on the IBI series, or the beat-to-beat intervals. And when we get to the IBI series and talking about it in a little more depth, we'll talk about what these statistics are and what they mean and how they're calculated, which is what we're going to do now. Now that we know what heart rate variability is and we know the signals that we're working with, we can talk about how to take those signals and make meaningful information out of them and obtain the statistics that we care about. And I'm just going to go through each of these processing blocks. These are the main steps that you go through from a raw ECG series to RSA and the other statistics sort of sprinkled throughout this. And I'm going to talk about each step and how we get there. And then after we're done with this, I'll open up the HRV application and we'll show you how to get that information using the application. So we're going to start with our raw ECG series, and we're going to apply some filters. So why would we filter our ECG series? Well, we're trying to obtain beat-to-beat timing. That's the root of studying heart rate variability, is to know when beats occurred relative to one another. And the way we're going to do that, as I mentioned, is by identifying the R peak on the ECG series. And we have some peak detection algorithms that do that. But the cleaner the signal and the more predictable the signal is that we pass to those algorithms, the more likely they are to correctly identify the R peaks. And that's going to result in less time needed to correct the mistakes that the algorithms make. So we have a couple of filters that we have available in the HRV application. The first one is the baseline and muscle noise filter, which is a bandpass filter between 0.5 and 45 hertz. And that's going to remove baseline drift. So if the signal's walking around at all, you're going to remove that. And then it's going to remove some high-frequency noise that's typically related to muscle activity. So we're going to isolate it to the frequency band that we know we care about for ECG when we're trying to identify an R peak. The next filter we have is the notch filter, which is a band stop at either 50 or 60 hertz. And this is used to remove electrical interference from the data. So whether your subject was coming in contact with a source of electricity, whether there are wires running to their feet that might be coupling this electrical interference onto the signals, this filter is going to remove that. And there's a toggle between 50 and 60 hertz because in North America, 60 hertz is the frequency at which electricity runs, and it's 50 hertz in Europe. So you have the ability to toggle between those. But that's what that filter is doing. And you really don't need to apply this filter always. It's best to look at your data, sort of examine it, see what sort of noise you're experiencing, and then apply these filters accordingly. But it's pretty much always safe to apply the baseline and muscle noise filter. You can just leave that on. Okay, so once we've filtered our ECG, we're going to now want to identify those R peaks on it and to derive this IBI series, or this series of interbeat intervals from which we can run some statistics and obtain, heart rate variability information. We use two types of peak detection in the Mindware application. The default is the Hilbert transform method, which is ideal for detecting R peaks on an ECG. You don't have to turn anything on for that to be used. It's going to automatically be done. We also have a manual override option, which is a basic threshold peak detection. You set a voltage level. Any peaks above that voltage level are going to be considered R peaks or beats. And this can be useful when you have high amplitude noise that's preventing some of the R peaks that you know are valid from being detected. And it can also be useful when you're working with PLE data. Now, the Hilbert transform method does work well with a lot of PLE data, but if you're having trouble with it, try using a manual override and you might get better results. Once we've identified our peaks, we now want to start looking at the intervals between those peaks. And we're going to transform our ECG series into something called an IBI series, which we're no longer looking at things in terms of seconds. We're not looking at a signal from 0 to 60 seconds, for example, in an IBI series. We're looking at a series of intervals where their durations are marked on the y-axis. So on this particular chart, we could see that the first interval starting around 800 milliseconds, we can tell from the ECG that the R peaks are getting farther and farther apart, and in the IBI series, we can see those durations increasing accordingly. And then as the heart rate begins to speed up at the end, we can see it begin to decrease. So that's the relationship between the IBI series and the ECG signal. Now from this IBI series, we can calculate a lot of statistics. Some of those include heart rate, which is reported in beats per minute. We calculate mean IBI in milliseconds, and these are going to be inversely related to your heart rate. So when your heart rate increases, the number of milliseconds between each beat are going to decrease. Therefore, your mean IBI is going to be lower, and vice versa. We can also obtain these time domain HRV metrics that I mentioned earlier. Some of the most common that are reported are SDNN, which is a metric of total variability. It takes into account all of these cyclical components that are contributing to variability. And RMSSD, or the root mean square of successive NN differences. Basically what we're doing is we're looking at the differences between subsequent intervals and taking the root mean square of that. And the reason that that statistic is particularly popular is because it's been shown to correlate highly with high-frequency HRV, or parasympathetic nervous system influence. One of the benefits of using time domain HRV statistics is that there's really only one way to calculate them. It can't vary from researcher to researcher. So if you want to compare results with other publications or other results, these are very safe to do that with because you can be confident that RMSSD was calculated the same way in all instances. That's not necessarily the case with RSA because there are multiple methods of obtaining RSA. We just happen to perform the frequency domain one in Mindware applications. The downside of using the time domain HRV metrics is that they've been shown to not be quite as accurate at portraying what they attempt to portray. So RSA is going to be a more accurate representation of parasympathetic nervous system influence than RMSSD is going to be. But we output them all because there are different places that each of these are useful. Okay, so now we have our IBI series. We have our series of interbeat intervals, and we need to start extracting some frequency information from them to isolate the frequency ranges that we care about and obtain RSA or our other frequency-based statistics. But in order to do that, we need to transform the IBI series a little bit. And that's dependent, the way we transform it is dependent on the requirements of the frequency analysis. We use a fast Fourier transform, or FFT, to transform the time domain signal, the IBI series, into the frequency domain. A couple of the requirements of an FFT are that the data needs to be evenly sampled, and there needs to be a sufficient number of samples, or data points, to measure the frequencies that you care about. In its current state, the IBI series has very few samples, and it's not evenly sampled. So we need to rectify those 2 points before we can pass it and transform it into the frequency domain. And we're going to do that by creating something called the heart period time series, which is an interpolated and detrended IBI series that's then ready to be transformed into the frequency domain. So we have our IBI series here, like we had on the last slide, and we're going to interpolate it based on 10 times the maximum expected heart rate, which is specified on the RPeak and artifact settings tab in the application, which I'll show you in a moment. So we're going to take the IBI series, we're going to insert data points between each of the known IBIs to create a smoother signal with more data points, and we're going to detrend it to remove that offset that's inherent in the IBI series, because we don't care about that offset when we're looking at frequency-based analysis. All that offset's going to do is interfere with our ability to measure more meaningful frequencies. So we're going to detrend it, interpolate it, and now it's ready for frequency-based analysis. And so finally, we're going to pass that to the FFT, and we're going to extract the frequency components from the very low frequency, and high frequency bands. Now, based on the age of your subject, there may be different appropriate band settings to use. Here I have listed some typical adult settings and some typical child settings, but they are highly dependent on age and the type of respiration rates that you're going to be seeing with that particular age group. And so if you want more information on how this is done, check out Knowledge Base Article 15 on our support site, and you get some more information on that. And finally, we can calculate RSA as the natural log of the power in the high frequency band, which I'm going to show you in a moment when we open up the application. So I'm going to go ahead and stop the presentation for now and bring up the HRV application so we can discuss that. I'm going to click on the HRV icon, which is what you see here. And the first thing that's going to happen when you bring up the application is it's going to ask you for a file. I'm going to navigate to where I know this file exists. And our applications are able to open a number of different files. There are the Mindware files, which are either .mw or .mwi if you're using newer Mindware equipment. We can also open acknowledge files if they're saved in AC3 graphs. And unique to HRV, we can open a text file that contains an IBI series directly, which I mentioned a little earlier. I'm going to start by opening up example1.mwi. And once you open your file, it's going to prompt you to map your channels. So you need to tell the application which channel you're using for ECG, which channel you're using for respiration, or if you're deriving respiration from cardiac impedance, you need to tell it which channels are the cardiac impedance channels. Now hopefully you've labeled your channels appropriately such that it's very obvious which one contains which signal. In this particular case, it wasn't labeled so well, so it just says Bio1, but I know that this is ECG based on how I acquired the data. I'm also going to be deriving respiration from the impedance waveforms, so I'm going to go ahead and select Z0 and DZDT. You really only need one of these. I'm going to be using Z0, but I'll map them both just to show you how to do it. Okay, once you've mapped your channels, you can press OK, and you're going to be on the setup screen. Now the first tab that you see is going to be events and modes, and this is where you tell the application how you want to view your data, how you want to segment it. And I'm not going to spend much time going over this today because it's sort of out of the scope of this webinar, but we do have another webinar that focused entirely on this tab and what the different modes do, and you can find that by going to our YouTube page. It's under the Events and Modes webinar. For today's purposes, I'm going to be looking at event mode with a post-event time of 60. And the one thing that I will mention while we're on this screen is the amount of time you should be looking at any given segment. So in order to obtain respiratory sinus arrhythmia, we need to look at a minimum of 30 seconds of contiguous data, meaning 30 seconds of uninterrupted, artifact-free data. And so we could set our post-event time to 60, but that starts to 30, but that starts to eliminate some of the editing possibilities that we have when we're cleaning our data. So to be safe, it's a good idea to look at things in 1 minute or 60-second segments. Moving on, you may recognize this screen from some of the screenshots in the presentation, but this is the HRV calibration settings tab. It's where you're going to tell the application how you're going to derive respiration. You can see that we don't have to derive respiration, but if you're reporting RSA, it's important to. But we can pick from either of the cardiac impedance signals, Z0 or DZDT, or we can choose a respiration source from a rest belt, for example. And below that, we have our frequency domain settings. We refer to them as the spectral analysis settings. And this is where you're going to be specifying the frequency bands that you care about for your particular population. So if you're studying an adult, these are the adult settings that are set in the application by default. But these are entirely programmable, and you can change them to work for different age ranges. Moving on to the next tab, we have the RPeak and artifact settings. So we're going to talk a little more about artifact here in a moment, but we'll recall from earlier the peak detection settings and the filters. So here's where you can turn on and off your filters and adjust your peak detection settings. And finally, under additional settings, if you want to view videos along with your data, you can map your videos here. If you already cleaned your data, you can select an edit file from here, which we'll also get to here in a moment. And you can specify where your Excel spreadsheets are going to be saved when you write your data out to them. So that's pretty much it for the setup screen. There's not really much you have to do here besides turn on some filters and adjust your frequency bands accordingly. And when you're ready, you can press the green Analyze button to proceed to the analysis screen. And the first thing that's going to happen when you go to the analysis screen is it's going to ask you if your ECG is inverted. So for the peak detection algorithms to work properly, we expect the RPeak to be rising in amplitude. And if you have an inverted ECG, maybe from swapping the leads on the body during acquisition, we're not going to be able to find that RPeak. We're going to end up identifying a Q wave or something like that as your beat marker, and it's not going to be as reliable. So you want to make sure that that RPeak is moving in the positive direction. And it's pretty obvious to tell when you're looking at a signal whether it's inverted or not. This particular signal is not inverted, so I'm going to press no, and we're going to be on the analysis screen. So here you can see all of the different processing blocks that I just described to you. So we have the filtered ECG across the top with peaks marked as either blue or yellow dots. And we'll talk a little bit about the yellow dots when we talk about data cleaning. Below that, we see the IBI series. And then below that, the transformed heart period time series and the power spectrum, or the FFT, thus showing the frequency content of the IBI series. And then finally below that, we see the respiratory time series. In this case, that's the derived respiration from impedance and the resulting respiratory power spectrum, which is the same processing that's done to the heart period time series, but done to respiration. And the reason we do that is we want to make sure that the peak frequency of respiration falls within our RSA band. And that's where the validation comes into play. So if you're planning on reporting RSA, it's expected that your respiration rate is within the high frequency band of frequencies. If it's not, you're going to see this respiratory peak frequency statistic on the right-hand side turn red. And in that case, you either need to look at your respiration time series and make sure there's not some sort of artifact or something going on, or you need to discard this particular segment because it can no longer be proven that your RSA is in fact valid. Additionally, on the right-hand side, we can see the other statistics that I mentioned, such as heart rate, mean IBI, and down below, the frequency domain statistics, such as the high frequency power, RSA. We output power for each of the frequency bands, which is essentially just the integral of that frequency band. And if you click on the other tab, you can see your time domain statistics, so SDNN, RMSSD. All these statistics are listed right here. So that's pretty much all you need to do to go from a raw ECG signal to all of these heart rate variability based statistics in the Mindware analysis application. One thing that we haven't talked about, though, is probably the most time-consuming portion of studying HRV and analyzing data, and that is data cleaning. So I'm going to jump back to our presentation. and let's talk a little bit about data cleaning and ECG editing. So we refer to errors in the data or anomaly in the data as artifact. And it's important for HRV that artifact be dealt with, or else it can have significant impact on your end result. And the place that we're going to deal with it is after peak detection, but before the IBI series is created. So we just saw that there are multiple colors of peaks. We have blue peaks and we have yellow peaks. Blue peaks are peaks that we think are good, that seem to meet the timing that we expect. And yellow peaks are peaks that may be artifact and may require some additional investigation and or correction. Now, ECG artifact can happen for a number of reasons. We could have movement noise, so noise from leads being pulled on or poor electrode contact during acquisition. So, and those sort of artifact are going to be very obvious. You're going to see sections of data that are almost non-recognizable. You won't be able to see R peaks necessarily in them at all. Or they could result in noise being identified incorrectly as an R peak. And the other thing that we care about in heart rate variability is the effect of arrhythmia or cardiac conditions. Because when we study heart rate variability, we're looking for variations in heart period, in beat-to-beat interval, that are caused by autonomic influence. We're not interested in the beat-to-beat variability that occurs due to some cardiac condition. So when these sort of things arise in our data, we need to correct them and restore normal beat-to-beat timing so that we're studying the right thing. We're not falsely identifying high variability for something that is really just an underlying cardiac condition. And that's one of the reasons that it's important to ask your subject what sort of known cardiac conditions they have, or if they're on any sort of medication that may be creating diminished variability. Now, the way that we identify artifact versus correct beats are by a couple of artifact detection algorithms. Specifically, the IBI min-max, which is just a threshold detector. It's going to look at, does your IBI fall above the maximum expected heart rate, or I guess below the maximum expected heart rate, or above the minimum expected heart rate? And then the MAD-MED, which detects outliers of beat-to-beat variability. And that algorithm is a bit more complex to go into right now, but if you check out the All About ECG blog series part 3, you can learn a little more about how that algorithm works, and it links directly to the publication where it was specified. So, as I mentioned, just having a few instances of artifact can greatly impact the RSA statistic. So let's take a look at this example. And in this case, RSA is 5.98. Now let's introduce just one artifact, just one missed R peak detection. Now we've increased RSA by an entire unit, which is significant statistically when you're studying RSA. And if you move to many artifacts, you can see that the impact is really tremendous on the end result. So it really is important to investigate each of those yellow peaks and deal with them accordingly. And what we're going to do now is jump back into our application and go over some techniques for dealing with different situations, different types of artifact. And it just so happens that the first segment in the example that we just had up has some artifact in it. We can see this section of yellow peaks, and we can also see in the IBI series this drastic change. So this sort of change from 700 millisecond IBIs down to almost 0 is not physiologically possible. It's not, you're never going to see that normally. So that can be a great indicator that something is definitely wrong here and we need to correct it. And we're going to correct it by using the ECG editor, which is accessible by pressing the green Edit Rs button. So I'm going to click that, and that's going to launch our editor. This is where you're going to spend most of your time when you're editing HRV data, or when you're analyzing HRV data. Go ahead and maximize this. And we do have another webinar that focuses specifically on this editor. It goes through every button in great detail. I'm not going to do that today. I'm going to just mainly talk about these specific instances and how to edit for heart rate variability specifically. But if you're interested in knowing every little bit about this editor, definitely go check that webinar out. So we can see here that there's a section of data that has yellow dots, and we're going to need to zoom into it a little bit to take a closer look. I'm going to do that by left-clicking down in the left-hand corner on this zoom tool and choosing the horizontal zoom option. I'm then going to left-click to the left of that area and drag across the area of interest. And when I release my mouse, you'll see that we've zoomed into that section. Now, in this particular case, we can see that this is definitely an R-peak. And we can see that this is definitely an R-peak. But in the middle, we had something that is most likely some sort of movement noise. And what we're going to want to do in this particular situation is delete these points, because we know that these aren't really R-peaks. These are just noise that the peak detection algorithm mistakenly chose as R-peaks. I'm going to do that by clicking the red delete button. And then when I move my mouse back into the field of the graph, you'll see that it's changed to an orange circle. This circle can be manipulated by using the mouse wheel. By scrolling up, you can increase the diameter of the circle, and by scrolling down, you can decrease it. And you can see at the top that the sensitivity bar is adjusting accordingly. So if you don't have a mouse wheel, you can always adjust the sensitivity using that as well. What this does is it gives you an area in which you're going to delete points. So if you're trying to delete something that is very specific point in time, you might want to make your circle a little smaller, but any points that are found within this circle when you click are going to be deleted. So I'm going to move my cursor so that the circle encompasses the point that I want to delete, and I'm going to use my left mouse, click, and it's going to remove that point. I'm going to do the same for this one. And when we do that, we can see that the points on either side of that noise have turned blue, because the IBI, the interbeat interval, now meets the timing criteria set forth by the artifact detection algorithms. Normal timing has been reestablished, and we can be comfortable moving forward with this data, knowing that it's clean. You'd want to scroll through the segment to make sure that there's no other instances of noise, and then press OK. And once we're back on the analysis screen, we can see that that same timing is reflected in the ECG series. That giant spike has been removed from our IBI, and this segment can be considered clean. At this point, you'd want to move on to the next segment. And we can see here that there is more noise. So I'm going to jump back into our editor and take a look. Again, I'm going to zoom in to that area so we can get a little bit of a better picture of what exactly is going on. And here there are a couple things. We can see that this is definitely noise. It doesn't look like a typical QRS complex or an R-peak. But we can see this point next to it that does look like an R-peak. It has the same characteristics as those around it. It just happens to be in a moment of movement noise. So the way I'd want to deal with this sort of situation is by deleting the obviously erroneous peak using the same method as we just discussed. And then I'd want to switch to my insert tool, which is this white button. And you're going to see the same orange circle. Again, whatever points are within this circle are going to be marked as peaks. So you can adjust the diameter of it accordingly. I'm going to position it so that the R-peak, or what we think is the R-peak, is within that circle. I'm going to click with the mouse, and we can see now that that was in fact the R-peak, because it restored normal cardiac timing, and the dots are now blue. Again, we can scroll through our segment to make sure that there are no other instances of artifact. If you want, we can come down here to the zoom tool and click the one in the bottom left-hand corner, which will zoom out to the full view. And we can see that all of our peaks are blue, and this segment is clean. Press OK. And again, it's reflected here, and we can consider this segment ready to go. Okay, now we're going to move on to the third segment, and we can see a different type of noise present here. So let's go ahead and open our editor and take a look. Again, I'm going to want to zoom in to the beginning of this segment so I can get a real good idea of what's going on. And what we have here is just exaggerated movement noise. So we have an entire section of data here that you really can't define well where the R peaks are. Recall something I mentioned earlier, that we need at least 30 seconds of contiguous data from which to get RSA. In this particular segment, we're looking at 60 seconds of data, which is beyond the minimum requirement to get meaningful information. So a technique that we can use in this situation to salvage this segment is simply to remove all of these peaks from the start of the segment. And that's going to be done by taking these blue vertical cursors. They're called the X cursors. And I'm going to drag one of them so that it's on the far side of the noisy section. And I'm going to drag the other one so that it's on the other far side of the noisy section. So these X cursors are bounding the area in which we can't really tell where the R peaks are. And I'm going to use a button labeled Delete All Points Between X Cursors. When I click that, they're all going to be removed. And this is okay to do because the remaining portion of the segment is greater than 30 seconds. So we can still get an idea of heart rate variability for this particular period of time, even after removing this small portion of data. Now, it's okay to do this from either end of the segment, so you can do it from the start or the end, as long as you end up with at least 30 seconds of data in the middle. But you can never do this in the middle of your segment, because then you're disrupting the contiguous R-peak series, and that gap in the middle is going to appear to the application as a very long IBI, and it's going to completely throw off your variability statistics. So you can always do this at the beginning and the end of the segments, but you can't do it in the middle, which is different than the way you would analyze for impedance cardiography, if you're familiar with that. So now that I've done that, I'm going to zoom back out. Everything looks good. I'm going to press OK. And at this point, the segment can be considered clean. So those are some pretty typical examples of noise that you'll see in ECG and how to deal with it. And there is one more that I want to show you, which is an example of an arrhythmia or a cardiac condition. So I'm going to press done and open a different file. But first, when you press done, if you've done edits to your data, you're going to be prompted to save your edit data file. And for HRV, this is with a .edh file extension. So you're always going to want to do this. It's going to save your data in an edit data folder in the same directory where your data is stored. So you want to press OK to save that file. If you ever want to recall your edited data, you need to first open the data file, the raw data file, and then come to the Additional Settings tab and choose your edit file from this Edit Settings section. This is done because it's possible to have multiple edit files for a single data file if you're working with multi-subject files. So you actually open your edited data by first opening the clean file, or by opening the raw file, and then selecting your edit file from this screen. So I'm going to open a second example quickly to show you how it looks to fix a cardiac condition or an arrhythmia that you encounter in your data. Now this particular file is dyadic, so it's multi-subject. This is one of those situations where you might have multiple edit files for a single data file. And I'm going to map the channels in the same way. So ECG, and we're going to be deriving from cardiac impedance again. So I'm going to select those two channels, press OK. And I'm going to jump right into the analysis screen so we can take a look at this. Okay. So again, we see that there are some yellow peaks here. We need to correct them. So I'm going to open the editor, and we're going to take a look. And again, I'm going to zoom in. So in this particular instance, all we need to do is delete it, and we'll restore proper timing. If we scroll out to this particular section, we can see there's not really any portion of this data that clearly represents an R peak. So the first thing to do is delete all of these points. And so now we have this gap in information. We have two yellow dots here, and there's most likely supposed to be a beat in the middle, but we can't clearly see one from the waveform. So we need to do something called a mid-beat, which is splitting the known IBI in half in order to reestablish normal cardiac timing. We're going to do that by dragging our blue cursors once again so that they are on either side of these known good R peaks. I'm going to turn off mid-beat auto peak. I'm going to press the blue mid-beat button. And we can see that it doesn't fall in the data on the actual waveform, but that doesn't matter because we're not concerned with the amplitude of the R peaks. We're concerned with the timing between them. So this is okay. And by doing this, we're going to estimate where the correct R peak would have been and restore timing, so now we can use this segment to obtain our statistics. Something important to note is that there's a limit to how often you can do this. So by doing this, we're sort of artificially creating data. We're guessing here. And that can ultimately have a negative impact on your end results. So it's ideal and recommended to limit the number of edits that you do in this way to 10% of the total number of R peaks in your segment. And it's better if you can do even less than that. So you want to aim to do as few of these as possible as to not skew your data, but 10% is sort of the limit that you can go to. So I'm going to exit the editor screen. We're back on the analysis screen. And we can see that all of the dots are blue, and our IBI series looks acceptable, and so this segment is done. If I want to write this out to an Excel spreadsheet, I'm going to press the white write button, and when I do that, it's going to output to Excel. And you want to do that for every segment in your file that you want to store the results for after you're done. So those are some examples of typical editing strategies that you can use in heart rate variability. Just remember that anytime you see a yellow peak, it's important to investigate it, take a look at what's going on, and correct it if necessary. And most times it will be necessary to actually make some sort of correction when you see that. Some additional resources, if you want to learn more about this, include the Handbook of Psychophysiology. It is the gold standard of many of the algorithms that we use in our applications. They're drawn from this book and related publications. And if you're interested in learning more about arrhythmias and how to recognize them and how to deal with them, this Goldberger's Clinical Electrocardiography is a really great book to have on hand. It's written for a cardiologist, so it does get pretty deep, but if you are running into a lot of subjects that have arrhythmias present in their data and you want to learn more about it, it's a great place to start. So with that, that concludes the presentation, the walkthrough portion. So if you have to exit, thank you so much for attending, and please do complete the survey on your way out. But at this point, we are going to move into the question and answer portion, start getting back some of the questions that you've had so far. So I'm going to take a moment and take a look at what questions have come in, and I'll be back here in a minute. Okay, so I have a question regarding arrhythmias, the topic that we just discussed, and maybe just asking to see another example of a different type of arrhythmia that you might see. So we pull back up that file and we go to the next segment. We could see something a little different than what we were seeing before, something that looks more like actual R peaks, but they're still being flagged as yellow dots. And I'm going to open up the editor to take a closer look. And I'm going to zoom in to the section with the yellow stars. So what we see here is something that actually looks like a QRS complex. We see the R peak. It's of relatively the same amplitude as the ones around it. But we can see that it's occurring much closer to the previous one than is typical. And in this situation, it is an arrhythmic beat. It is not normal cardiac timing or variability that's been created by autonomic regulation. So even though this is an R peak, it's a perfectly valid R peak, we don't want to analyze using this R peak because the variability that we're seeing here is not from autonomic control. It's from the cardiac condition. So much in the same way as we did before, we want to take our blue cursors and position them on either side of the yellow dots and insert a midbeat. And we can see now that the cardiac timing has been restored. So even in situations where there's an arrhythmia that occurs where it's clearly an actual R peak, in this case, you still want to correct for it. And this does count towards the 10% of total corrections that you can make in a given segment. All right, we have another question regarding deleting data from the start of a segment. So I'm going to jump back over to that file, save my edit that I just did, and I'm going to open the first example that we talked about. And I did not load my edit file, so this is going to be raw data. And I'm going to go to that particular segment. So I was actually only selecting a very small amount of data from this window. So if I zoom back in here to the area of interest, and we look at our x-axis, we can see how much time has elapsed when we're selecting data. So if I'm going to select that same area again by taking my blue cursor and moving one to the front and one to the end of that section of data, we're actually only looking at about 4 seconds of data, which is completely acceptable to remove that information and still use the statistics from this particular segment. So I'm going to go ahead and delete those points again and press OK. And we can see that that's actually a relatively small amount of data that we've deleted. Okay, so we have a question. If you didn't record respiration and you don't have impedance, can you still use RSA? And the answer is technically you can. You don't need respiration to get an RSA value. It's entirely calculated from the IBI series. So the actual respiration waveform and the resulting statistics don't actually influence the calculation of RSA. But what you do need is proof that your subject was breathing at the rate in which RSA can be calculated. It needs to fall within that high-frequency RSA band frequency. So if you have too many breaths or too few breaths in a particular segment, it may invalidate RSA. So from a reporting perspective, from a publication perspective, it's important to have respiration, but technically you can calculate RSA without having a respiration waveform. There's a question regarding inversion of respiration. So it doesn't matter if the polarity is swapped on your respiration signal. And the answer is no, it doesn't matter at all. We can still derive rate from respiration, whether it's increasing with inspiration or increasing with expiration. Okay, there's another question regarding the need to remove all yellow peaks in order to have a valid analysis. So the yellow peaks are just peaks that have been identified by the artifact detection algorithms as potentially problematic. That doesn't mean that they are problematic. So you really just need to examine your data and look closely at the peaks to determine whether there's just an exceptional amount of variability at this particular point in time, or there's some sort of arrhythmia, in which case that needs to be dealt with, or if the R peak has been erroneously identified or missed, at which point you need to either insert or delete that R peak. So it's okay to have some yellow stars if you're comfortable with the waveform morphology and if you're comfortable with the timing of the R peaks. All right, we have a question regarding the difference between deleting all points and deleting a portion of data. So what I did here is delete the points. So we can still see the waveform, but we remove the R peaks. And since the R peaks are what are driving the IBI series and all the subsequent calculations, that's all we really need to do to edit our data. But we have another option, which is remove data portion. And what that's going to do, if I were to perform a similar operation here, instead of deleting all points between X cursors, I could press the orange remove data portion button. And that's going to zero the data. There's not one way that's necessarily better than the other because they're both going to have the same end result of removing those erroneous R peaks from the IBI series and the resulting calculations. What you lose when you remove data portion is any visual indication of why the points were removed from that period of time. So when you do this, it just looks like a flat line. So a person that's reviewing edits or looking at this data later can't obviously tell what happened here. Now, of Of course, the edit data file, or the edit file, is separate from the data file, so your raw data is always preserved. They can always unmap the edit file to look at what this waveform used to look like and then understand why you removed it. But if you just delete all the points instead, you still have this visual record of this is what the signal looked like, so that a reviewer can say, obviously you can't identify R peaks here, this is why they did that, and this segment is okay. So that's why I would typically recommend just deleting the points as opposed to removing the data portion. Okay, we've also got some other questions about arrhythmias, and it's definitely a complicated subject. So if you look at the All About ECG blog series, we do have an entire post that's dedicated to talking about arrhythmias, how to identify them, and how to deal with them. Specifically, there are some questions regarding multiple arrhythmias within a segment, or when they have what's considered a reset of the cardiac timing. Some arrhythmias will result in a change in cardiac timing such that after the arrhythmia, the cardiac timing is different than before it. And that's particularly problematic for heart rate variability analysis because it can potentially impact large portions of data within a segment. And so when you have a reset, the best thing you can do, and ideally the reset occurs near the beginning or near the end of your segment, and you can just delete all of the data points on one side of the reset. So therefore you're only looking at one section of cardiac timing. If you can't do that, then you're most likely not going to be able to use that segment of time. If you have multiple arrhythmias, it's perfectly fine to go through and correct them all, just as I had shown you, with the midbeat function. And you can do that up to the 10% rule, or 10% of your total number of beats in a given segment can be estimated with the midbeat. If you go any higher than that, it starts to skew your results in such a way that it's difficult to really rely on them. And so 10% is sort of the cutoff, but you really want to keep it even lower than that if possible. There's a question about how to tell the difference between yellow dots that are due to arrhythmia versus just very high variability. Well, the best way you could do that is to look at the morphology of the QRS complex. So most of the time when you're dealing with arrhythmias, you're going to see a beat that mostly looks right, but something about it is not going to quite look right. Whether that is a widened QRS complex or the absence of a P wave, which is the wave that precedes the QRS complex. I can show you that if I go back to our slide. So if we look at the traditional landmarks that are seen on a ECG signal, again, we can look at how these landmarks are different from the typical QRS complex or the typical ECG morphology. If you see any abnormalities in your ECG cycle, then it's most likely arrhythmia and you need to deal with it. But there are times when there's just high variability and it just barely exceeds those limitations of the artifact detection algorithms. And in that case, it's okay to leave it. Okay, we have another question regarding deleting the peaks in the noisy segment and what the benefit is of deleting the R peaks versus leaving them as yellow artifacts. Okay, so I'm going to exit out of here and I'm not going to save my edits. So we can look at this again as a raw waveform. So my signal is not inverted, and I'm going to go to segment 3. Okay, so here, take a look at the IBI series. We can see that we have IBIs down here at nearly 0 milliseconds, so many points that are very close to one another. And it's clearly not something that is real. These aren't real beats. These are artifacts. These are incorrectly marked R peaks due to noise. And as a result, our heart rate variability statistics are going to be off considerably in this particular case because there are so many erroneous peaks being marked here. So it's entirely unacceptable to leave these peaks as yellow. We need to remove them, or else we're going to be looking at variability due to noise, essentially. So let's go in and one more time delete these peaks from the beginning of the segment. Exit out this real quick. Zoom in so I can get a better view of the data. I'm going to move, again, the cursors on either side of the noise. Recall that our RSA right now is at 4.85. I'm going to delete all these points. And now our RSA is at 3.67. So a significant change in RSA due to the noise that we needed to remove. So that's really why those yellow peaks need to be taken care of. Okay, so it looks like questions have stopped coming in, so we're going to go ahead and wrap up the presentation. Again, thank you so much for attending, and please do complete that feedback survey as I mentioned before. And if you have any questions, definitely refer to our support site, as it is always growing, and you can expect to see a blog series specifically on heart rate variability coming up in the next few weeks. So thanks again, and have a great day.